Good afternoon. My name is Cassie Starnes, and today we'll be talking about the Watson and Crick structure of DNA. The first thing to talk about is base pairing. DNA bases pair exclusively with one other base. Adenine will only pair with thymine in DNA, and cytosine will pair exclusively with guanine. Adenine and guanine are purines, whose structure is seen here as the double ring, while cytosine and thymine are pyrimidines. Pyrimidines are formed by a single ring and will bond with hydrogen bonds to their complementary purine. Base stacking. Base stacking is the general arrangement of DNA bases in the double helix structure. The base pairs are flat, as seen here represented by the cardboard. These bases are arranged perpendicular to the phosphate backbone. This arrangement maximizes hydrophobic interaction, which stabilizes the DNA. Hydrogen bonds connect the complementary bases in DNA. Guanine and cytosine form 3 hydrogen bonds, as represented here by that string, and adenine and thymine form 2. Hydrogen bonds are relatively weak between positive and negative charges, and they are easily broken. The phosphate backbone. So the phosphate backbone provides structure for the DNA double helix. The backbone is formed by a sugar and phosphate group alternating, as shown here. The phosphates are bound to each other by phosphodiester bonds. The sugars of the DNA backbone are formed from 5 carbons, also shown here. The carbons are designated by number depending on when their base is attached. These numbers indicate the direction of the strand. The first carbon is bonded to the base, while the second carbon is bonded to a hydrogen group in DNA, but an OH group in RNA. The 5' carbon is bound to the phosphate group, also shown here. Whether the 3' or the 5' carbon is left unconnected to another nucleotide determines the direction of the strand and determines the direction of replication. Major and minor grooves. The major and minor grooves refer to the spaces between the phosphate backbone as it twists in the double helix structure. Minor grooves are the spaces between the 2 strands as they curve together, shown here, while the major grooves refer to the larger gap between the DNA backbones as they align with each other at different points in the DNA structure, also shown here as the DNA twists behind where we can't see it. Handedness. Eukaryotic DNA is exclusively right-handed. As you see here with the purple line representing the backbone, it's curving up and to the right, meaning that this is right-handed DNA. Some examples of left-handed DNA in the media are these 3 pictures. This Backstreet Boys DNA album cover shows the stairs curving up and to the left. This DNA Destination Management logo shows this gray DNA curving up and to the left. These lights, which are representing a DNA double helix, are curving up and to the left as well. These other 2 pictures, so the studio DNA curves up and to the left if you focus on this orange strand, or you turn it the other way, the pink strand. And this news article about the Trump administration also shows it curving up and to the left. These are my sources. Thank you very much for your time. Have a lovely rest of your quarantine.